So today we will learn the Chapter 5, Epigenetics in Mammalians. So Chapter 5 is composed of 7 subjects starting from DNA methylation and DNA demethylation and non-coding RNA and also chromatin insulator. Finally, we will cover super enhancer. So in Chapter 1, we learned the concept of DNA methylation. So in this slide, I will go over DNA methylation again. So DNA methyltransferase DNMT induce methylation at cytosine. So this is cytosine, and this is first, second, third, fourth, fifth, and sixth location. So always in fifth location can be the methylated. So it will be 5-methylcytosine. So DNMT1 and DNMT3A, DNMT3B, all of them induced DNA methylation, but they induced DNA methylation a little bit differently. Okay. For example, DNMT1 is a recreate methylation pattern on hemimethylated DNA. So this is the hemimethylated DNA. One strand is methylated, the other strand is not methylated. So in this case, DNMT1 recreate the methylation pattern on the hemimethylated DNA. So this is the role of the DNMT1. So recreate methylation pattern on hemimethylated DNA. How about DNMT3A and 3B? So they did de novo methylation. So de novo methylation. So initially, you see here, there was no methylation in both strands, and DNMT3A and DNMT3B newly synthesized DNA methylation in both strands. So this is, we call it as the de novo methylation. So in this slide, I will explain the first subject overview of DNA methylation. Okay, DNA methylation is a process by which methyl groups are added to DNA molecules. For example, this is cytosine. So cytosine have the first, second, third, fourth, fifth location. So fifth location is the methylated. So cytosine is become the 5-methylcytosine. So we also have to know the concept of CpG island. Because the CpG islands contains a large number of CG dinucleotide repeats. So it means that a cytosine base followed by guanine base. Okay, CG, CG, CG. So in such an arrangement, the cytosine in such an arrangement tend to be methylated. So this is the CpG island in the genome. Okay, and you will see that CpG islands occurs at, this is the transcription initiation site. So CpG islands occurs in everywhere in the genome, but CpG island is also occurs at or near the transcription start site of gene. Okay, so if CpG islands is unmethylated, so transcription can be initiated. So when the CpG island is methylated, so transcription is abolished. Okay. So in this slide, I will explain the classical maintenance methylation model. So there are two models. First one is conservative DNA methylation. Second model is de novo DNA methylation. conservative DNA methylation means that recreate methylation pattern on hemimethylated DNA by DNMT1. Okay, so recreate, so this is hemimethylated the DNA, so recreate the methylation pattern, so this is the hemimethylated DNA, recreate the methylation pattern on DNA by DNMT1. So we call it as this is the conservative DNA methylation. Second model is de novo DNA methylation. So de novo DNA methylation means that newly synthesized DNA methylation. So there was no methylation here, but after DNMT3A and DNMT3B functions and induce methylation, so it will make the newly synthesized DNA methylation on both strands. So this is the de novo methylation. So in this slide, I will explain important concept in classical maintenance methylation model. So this is prediction. The methylation status of a single CPG site are stably inherited. Okay, for example, so this cell become this cell, and this cell's offspring will be this cell, and this cell's offspring will be this cell, and this cell's offspring will be these cells. Okay, in this case, is all of cell has the same methylation status of a single CPG site. But however, so if so, and also a perfect maintenance enzyme for this task exists. If so, all cells on one tissue, in one tissue they have a different cell. All cells of one tissue have same methylation status of a single CPG site. So all cells of the one tissue will have the same methylation status of a single CPG site. However, we found that all cells of one tissue have a different methylation status of a single CPG site. All cells have a different methylation status of a single CPG site. However, the average methylation density are maintains. So this is the one cell, this is one cell, and this is another cell. Okay, so we found that this is the distance from the nucleosome. For example, 0 and 200 base pair, and 400 base pair, and minus 200 base pair, and 400 base pair. And this is the average methylation density of one cell, and also this is average methylation density of one cell, and we found that these cells have similar average methylation density. So it means that average methylation density are maintains. So the conclusion is that average methylation density are maintains, but are not exact CPG site-specific methylation pattern. So this is the very important concept in the classical maintenance methylation model. In this slide, I will explain the mammalian DNA methyltransferase structure and function and again. So this is the DNA methyltransferase. Okay, they have two specific domains. First one is the regulatory domain, and the other one is catalytic domains. For example, DNMT1 has the both domain. However, DNMT2, they don't have the regulatory domain. They only have a catalytic domain. And DNMT3A and 3B, they have both domain, regulatory domain and catalytic domain, but they have different domains like this. Okay, they have PWWP domain and also they have ATRX domain. Okay, the function of DNMT1 is recreate methylation pattern on hemimethylated DNA, but the DNMT2 is no specific function is known, and also DNMT3A and 3B, they can do de novo DNA methylation. So they can induce newly synthesized DNA methylation. Okay. So in this slide, I will explain the cooperative activity between DNA methyltransferase. Okay, so this is a wild type cell. Wild type cell has the methylation pattern in the CpG island. Okay, so most of the CpG island is methylated. However, when DNMT1 is knocked out, we found that DNA methylation decreased. Also, when DNMT3A was knocked out, we also found that DNA methylation decreased. And also, when DNMT3B is knocked out, we also found that DNA methylation is decreased. But when DNMT1 knockout and the DNMT3A is knocked out, when the double knockout happens, we found that DNA methylation is further decreased. And also, when DNMT1 and DNMT3B was both of them is knocked out, we also found that DNA methylation is further decreased. So based on this observation, the conclusion is that DNMT1 and DNMT3A or DNMT1 and DNMT3B are cooperative to maintain DNA methylation. So this is the cooperative activity between DNA methyltransferase. So in this slide, I will explain the bisulfite sequencing that identifies DNA methylation. So before DNA sequencing, DNA was treated with bisulfite. Actually, the full name is sodium bisulfite. After that, this bisulfite treatment converts cytosine to uracil. However, but leaves 5-methylcytosine residues unaffected. So this is an important thing. So bisulfite treatment convert cytosine. So this is the cytosine. After cytosine is converted to cytosine sulfonate. After cytosine sulfonate can be changed to the uracil sulfonate. Finally, the uracil sulfonate is changed to the uracil. In cytosine and uracil, their structure is nearly similar to each other. Okay. However, bisulfite treatment leaves 5-methylcytosine unaffected, still 5-methylcytosine. Okay. So after bisulfite treatment, cytosine will be converted to uracil and 5'-methylcytosine will still be 5'-methylcytosine. So during the PCR reactions, U will bind to the adenine, uracil will bind to the adenine. So after PCR reaction, after one more PCR reaction, adenyl will bind to thymine. So finally, uracil will be changed to thymine. Okay. So after sequencing, we can observe thymine. Okay. However, methylcytosine region, so after PCR amplification, So methyl cytosine region will be the cytosine, and after sequencing it will be the cytosine. Okay, so this cytosine is not unmethylated. This cytosine is methylated. So unmethylated cytosine will become the thymine. However, methylated cytosine will still be C, cytosine. So after DNA sequencing, cytosine, this is cytosine. Cytosine stands for methylated cytosine. It's okay. So in this slide, I will explain the transcriptional regulation by DNA methylation. So usually the DNMTs make the induced DNA methylation, especially in CpG islands. After that, DNMT forms complex with histone deacetylase 1 or 2, or methyl CpG binding protein MECP2 or MBD1, 2, 3, or it also complex with tumor express gene. After that, it can induce transcriptional repression. Okay, this is the one mechanism. Another mechanism is that this is the MECP2. MECP2 is the methyl CpG binding proteins. Okay, methyl CpG binding proteins, MECP2. MECP2 has the MBD domain and the TRD domains. This is MBD and TRD domain. The function of MBD domain is this is the MECP2. MECP2 has the MBD and TRD domain. MBD domain is bind to the methylated CpG islands, and also TRD domain is bind to the histone deacetylase. After this binding happens, it will induce transcriptional repression. Okay, so there is two mechanisms. One mechanism, DNMT itself forms complex with the HDAC or methyl CpG binding proteins or tumor suppressor. So it will induce transcriptional repression. Or MECP2 itself has the MBD and the TRD domains. After it binds to the CpG islands, after that it will recruit HDAC. After that there will be transcriptional repression. So in this slide I will explain the Rett syndrome. So in previous slide we learned that MECP2. So this is MECP2 has the MBD domain and the TRD domain, and they also have several other domains. So MECP2 is located at X chromosome. You find that there are several mutations can be happens in the MECP2 gene, and each mutation is causes cranial nerve disorder. So this is the cranial nerve. We call it as a 내신경. Cranial nerve is composed of several the nerves. Okay, so cranial nerve disorder. So finally, it causes mental retardation in women. So what is Rett syndrome? The most severe form of autism. Autism is a mental retardation that only affects girls. Okay, so if the mutation is happen in MECP2, it causes the cranial nerve disorder, and finally it causes mental retardation in women. So in this slide, I will explain the second subject, DNA demethylation. So what is DNA demethylation? DNA demethylation means that removal of methyl group from nucleotide. So this is 5-methyl cytosine, finally it will become the cytosine. So this step we will call it as DNA demethylation. So two enzymes is involved in the DNA demethylation. First one is the TET, 10-11 translocation. The other name is the other enzyme is TGD, thymine DNA glycosylase. Okay, let's see some detailed function of each enzyme. Okay, the main function of TET is that they induce oxygenation. For example, this is the 5-methyl cytosine, and after TET is involved in this step, they will change it to 5-hydroxyl methyl cytosine. 5-hydroxyl methyl cytosine, hydroxy. After that, they will induce the oxygenation once again. They will make the 5. So they will make the hydroxyl group to aldehyde group. So they will make the 5-formyl cytosine. So finally, TT induced oxygenation again. Finally, they will make 5-carboxyl cytosine. Okay. After that, TGD involved in the final step. Okay, TGD is making the cytosine, finally making cytosine from 5-formyl cytosine or from 5-carboxyl cytosine. Okay, so they will change it, they will remove this group or they will remove this group. Okay, so once again, DNA demethylation is the removal of methyl group from nucleotide. Two enzymes, TT and TGD, involved in the DNA demethylation. So actually, we have to memorize this step because this is very important procedure in this chapter 5. Okay, 5-methyl cytosine be oxidized and it will make the 5-hydroxymethyl cytosine, and it will be also oxidized. After that, it will make the 5-formyl cytosine, and after that, it will be also oxidized and it will make the 5-carboxyl cytosine. And TGD is involved in the final step to make the cytosine. So they change it 5-formyl cytosine or they change it 5-carboxyl cytosine into cytosine. It's like this. Okay, so we have to memorize DNA demethylation procedure. Okay, so in this slide I will explain the DNA demethylation and the methylation mechanism during development. Okay, so this is the x-axis, means that embryonic stages. Okay, and y-axis is CpG, the percentage of CpG methylation. Okay, so this is the sperm and this is the oocyte. So sperm and oocyte can be fertilized. After that, the zygote will become blastocyst. Okay, so in these cases, test 3 is working. So test 3 induces demethylation from fertilization to blastocyst stages. So demethylation is happen. Okay, after that, blastocyst will become the epiblast. Okay, so during these stages, the DNMT3A and DNMT3B induces de novo methylation from blastocyst to epiblast. The embryonic stages is an epiblast is E6.5. Okay, so around here, primordial germ cell can be made here, around here. So if you see here, so this is the primordial germ cell. After that, primordial germ cell can be seen here, around here. After that, primordial germ cell can be migrated in downstairs. After that, the primordial germ cell will go upstairs. Finally, it will settle down here. So this is the primordial germ cell. The embryonic stage is about E13.5. Okay, here. Okay, in this during these stages, TET1 and TET2 is function. So TET1 and TET2 induces dematuration from epiblast to PCG, primordial germ cell. Okay, during these stages, so dematuration happens by TET1 and TET2. Okay. So in this slide, we will learn the third subject, long non-coding RNAs. Previously, we learned short non-coding RNA. Short non-coding RNA is a type of RNA. The length is around 20 to 31 base pairs. So short non-coding RNA is composed of the microRNA, miRNA, and snRNA, and snRNA, and also tRNA, and finally there is the piRNAs. In the case of the free miRNA, microRNA, and snRNA, snRNA, tRNA is usually processed by dicer, so it will become to 20 to 30 base pair small RNAs. However, the piRNAs have a precursor. After that, Zook bind to the antisense transcriptor. After that, trimer and HANT is making the 26 to 31 base pairs piRNAs. So this is the this is a short non-coding RNA. Okay, once again, the length is 20 to 31 base pair. Is composed of the miRNA and snRNA and snRNA and tRNA and piRNAs. Finally, they will become the around the 20 to 31 base pair RNAs. Okay, so long non-coding RNAs, we call it as LNC RNA, is a type of RNA. Its length is exceeding 200 base pairs. Okay, there are three types of the LNC RNAs. First one is the intergenic non-coding RNAs. This one is located between, this is one gene, and the other gene is located here, located between genes. Another long non-coding RNA is intragenic. So this is exon and this is exon. So intragenic non-coding RNA is located between the exons. So it will become this one will become sense, and this one will become antisense intragenic long non-coding RNAs. Another form is the enhancer RNA. So this is eRNAs. So eRNAs is transcribed from DNA sequence of enhancer. So this is the enhancer region. So usually this eRNA is bidirectional. Okay. So once again, long non-coding RNA is composed of usually the length is exceeding 200 base pairs. They are the intergenic or intragenic or enhanced RNAs like this. Okay. Cut is the one example of long non-coding RNAs. Okay, we call it as cryptic unannotated transcript. So cut is the non-coding RNA that are produced from intergenic and intragenic region. Okay, usually the length is less than 1 kb. So they have the 5' cap, and they have the poly AAA tail, but not full length. Okay, so actually the poly A site is located here. Okay, they just have the small AAA, and usually this cut long non-coding RNA cut is this long non-coding RNA is rapidly degraded by exome. Okay, because their structure is not stable, because they only have the AAA site. Okay, so because they are unstable, so usually this cut long non-coding RNA is usually rapidly degraded by exome. However, this is SUT. SUT. SUT is the stable, uncharacterized transcript. So this is the long non-coding RNAs that are produced from intergenic region. And just the size is larger than 1 kb. So this is poly A site. So they passed poly A site. So they have the 5' cap, and also they have a poly A tail. So because they are full length of the poly A tails, so they are not degraded by exome. They are just degraded by decapping enzyme DCP1 and DCP2, and or the cytoplasmic exonuclease XRN1. Okay, so this is another type of the long non-coding RNA, SUT. The full name is stable, uncharacterized transcript. Okay, so in this slide, I will explain the transcriptional regulation by long non-coding RNAs. There is two mechanisms. One mechanism is a cis-acting regulation. The other one is a trans-acting regulation. First, cis-acting regulation happens. So LNC RNAs modulate the activity of a surrounding gene on the same chromosome. So long non-coding RNAs, this one can modulate the activity of the surrounding gene in the same chromosome. Okay, so this is recorded as cis-acting in the same chromosome. However, the trans-acting means that long non-coding RNA is transported to another chromosome. After that, modulate the activity of a certain gene in other chromosome. So this is recorded as the trans-acting regulation. This is the chromosome A, and this is chromosome B. So this is the long non-coding RNA is transported to other chromosome. So it will do trans-acting regulation. So usually this is the nucleus, and for example, this is the chromosomal territories within nucleus. For example, chromosome 1, chromosome 2, chromosome 3, 4, 5, 6, 7, 8, 9, 10, like this. So usually the chromosome, occupying the nucleus, physically approach with each other. They are physically approach with each other. So because of this configuration, that it can modulate the activity of a certain gene in other chromosome. For example, this the long non-coding RNA in this chromosome, because this chromosome is contact with this chromosome, so it can modulate the activity of certain gene in the other chromosome. So in this slide, I will explain the one example of transcriptional regulation by cis-acting long non-coding RNAs. So we learned that eRNA is transcribed from the DNA sequence of enhancer region. This is eRNA, so they can be transcribed in bidirectional manner. Okay, and after that, we also have to know another transcription factor. This is the NELP, is another transcription factor which prevents the progression of RNA polymerase II in the DNAs. So it means that NELP prevents transcription initiation or transcription elongation. However, the eRNAs, the transcribed eRNA removes the NELP. This eRNA can remove the NELP and can induce its transcription initiation and elongation. So eRNA is function as a cis-acting long non-coding RNAs, so it can modulate the activity of surrounding genes on the same chromosome. So this is the transcriptional regulation by cis-acting long non-coding RNA. So in this slide, I will explain the transcriptional regulation by trans-acting long non-coding RNAs. Okay, so one example is hot air. So this is hot air. So this long non-coding RNA is located HOX-C gene cluster. So this is the one HOX-C gene, and this is one HOX-C gene. So HOX-C gene is located as a cluster. Okay, so because the hot air is located between one gene and the other gene, so we call it as an intergenic long non-coding RNA. So hot air is located in chromosome 12, and this long non-coding RNA is highly transcribed in the cancer cells. Okay, so this is normal cell and this is cancer cell. And also hot air makes complex with PRC2. In previous chapter, we learned that what was PRC2. So PRC2 has the EZIT domain. So EZIT domain has the H3K27 methyltransferase activity, and they have ESC domain. They recognize the H3K27 methylation. So their main function is causes facultative heterochromatin in higher eukaryote. Okay, so this complex is bind to the HOX-D gene in chromosome 2. So, okay, hot air is located in chromosome 12, but HOX-D gene is located in chromosome 2. Okay, so these complexes bind to the HOX-D gene in chromosome 2. After that, PRC2 has the H3K27 methyltransferase activity, so H3K27 methylation. So finally, it will induce gene silencing in HOX-D genes. So the conclusion was that hot airs modulate the activity of HOX-D gene in other chromosome. So this is the transcriptional regulation by trans-acting long non-coding RNAs. In this slide, I will explain the fourth subject, the chromatin insulator in mammalian. So what is chromatin insulator? Chromatin insulator is a regulatory element that can bind to the DNA. So this is chromatin insulator, 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 insulator. So it has three functions: barrier, enhancer blocker, and insulated domain. Okay, let me start barrier. Okay, so this is our gene of interest, which is composed of the enhancer and promoter. After that, our gene of interest can be actively transcribed. Okay, so next to our gene of interest there is the heterochromatin region is located here. Okay, if insulator is located here, it means that it functions as a barrier. It prevents heterochromatin spreading. Okay, so that's why our gene of interest can be actively transcribed. Next function is the enhancer blocker. So this is the enhancer and this is promoter. Between the enhancer and promoter, the insulator is bind to here. So in this case, it prevents enhancer from interacting with promoter. So in this case, there will be no transcription. The final function is it can function as insulated domain. Okay, this is our gene of interest, which has enhancer and promoter, our gene. Okay, so first placed insulator in both sides of the gene. Okay, in this case, it can generate a functionally independent domain. This is functionally independent domain that can be protected from the neighboring region. So this is, we call it as insulated domain. In this case, our gene of interest is actively transcribed regardless of the neighboring region. So this is the insulated domain. Okay. So in this slide, I will explain the CTCF. This is a CCC-TC binding factor. So CTCF is chromatin insulator in the mammalians. So CTCF is not found in the yeast, worm, and flange. And that's why the CTCF is chromatin insulator in the mammalian. So they have the zinc finger domain. So this is zinc finger domain, and this is zinc. Okay, so this is they have the zinc finger domain. So this domain can bind to the DNA region. So they have it. So that's why they can. This is the CTCF. They can bind to the consensus sequence. Okay, so CTCF can bind to the consensus sequence, which is a repeat or core sequence, that's CCC-TC. Okay, they bind to the consensus sequence, but this consensus sequence is a variable. Okay, like this. Okay, for example, CTCF can bind to some specific consensus sequence, which is for example, in this case is GCATCC, a little bit variable, isn't it? And also another case is the CCA or CCACC, something like this. There's another variable is that CC, another consensus sequence is GGTGG is like this. Another variable is the CACTCA like this. Okay, so CTCF can bind to the consensus sequence. This is the mainly repeats of core sequence CCTCC, but this sequence is a little bit variable. It's like this. Okay, and also neighboring. So this is the one CTCF, and this is another CTCF, and neighboring CTCF interact with each other. Okay, and also we found that knockout of CTCF result in the early embryonic death it means that CTCF plays important role in the embryonic development. Okay. So in this slide, I will explain how does CTCF works. Okay. So this is the green is CTCF, and this region is CTCF binding site. Okay. CTCF bind to the CTCF binding site. After that, another protein cohesin is also bind to DNA, and so this is the actual structure of cohesin. So the cohesin can bind to the DNA, and it can forms the chromatin loop is like this. So cohesin bind to DNA, so it forms chromatin loop is like this. After it forms chromatin loop, so neighboring CTCF can interact with each other. At that, finally it will make the insulated domain is like this. So it can make the insulated domain is like this. They will make the insulated neighborhood. They can make insulated domain. Okay, so there will be two cases. For example, this is insulated domain. So in this case, enhancer and promoter is located within insulated domain. So in this case, it enhanced the interaction between the enhancer and promoter. It can activate transcription. However, there will be another case. For example, enhancer and promoter. This is enhancer and promoter can be separated by enhancer is located in the neighborhood, but the promoter can be located in the within the insulated domain. So in this case, it blocks the interaction between the enhancer and promoter. It can inhibit transcription. So this is the CTCF. I explained how does CTCF works. Okay, once again, CTCF bind to the CTCF binding site. After that, cohesin also bind to the DNA, so it forms chromatin loop. So in this case, neighboring CTCF can interact with each other. So finally, they will generate insulated domains like this. Okay, so there will be two cases. One case is they can activate transcription. Another case is it can inhibit transcription. One case is that enhancer and promoter is located within insulated domain. So in this case, enhance the interaction between the enhancer and promoter. It can activate the transcription. But when the enhancer and promoter is separated by the CTCF, in this case is block the interaction between enhancer and promoter. It will inhibit transcription. So in this slide, I will explain the fifth subject, TAS. TAS is topologically associating domain. So TAD is the self-interacting genomic region. So TAD is composed of the repressed TAD compartment and active TAD compartment. Let me start repressed TAD compartment. Okay, now this figure to show that chromosome territories within the nucleus. For example, chromosome 1 is located here, and chromosome 2 is located here, chromosome 3 is located here. So if we magnify this region, okay, so this region is this region, okay, so there is the nucleus membrane is outside, Okay, and next to the nuclear membrane, there is the nuclear lamina is located here. So this is nuclear membrane, and next to the nuclear membrane, there is nuclear lamina. In Korean, we call it as a 핵청. Okay, so this region, we call it as the repressed TED compartment. So usually in this region, chromatin bind to the nuclear lamina. So there are lots of the interaction with the chromatin and the nuclear lamina, chromatin and nuclear lamina. So in this compartment, interaction between chromatin is repressed. That's why we call it as a repressed TED compartment. How about active TED compartment? So this region, we call it as active TED compartment. So in this compartment, interaction between chromatin is very active. Okay, so usually the promoter or the enhancer are concentrated in this region. For example, this is gene. Okay, gene has each promoter or enhancer. So in TED, active TED compartment, there are lots of the regulatory elements. We call it as promoter and enhancer. The promoter and enhancer is concentrated in this region, so they can interact with each other like this. Okay. So in this slide, I will explain the heat map of TED. So this is a heat map of TED. Heat map of TED shows the interaction frequency between the chromatin. Okay. So interaction frequency is expressed as a color. So dark red means that interaction between this chromatin and this chromatin is high. If they show that the blue color, it means that interaction around this position in chromosome and this position in the chromosome, interaction between here and there is not nearly happens. Okay, how about the pink color? Pink or the pink color means that interaction is a little bit medium. Okay. Okay, so let me start the contact enrichment region. Okay, so this is the chromatin structure. Around this is one compartment, and this is one compartment, one compartment, one compartment, and one compartment. Within this compartment, so self-interacting is highly actively happens. Okay. So let me start this compartment. Okay, about around this region and this region, between this and this region. So maybe the interaction is actively happens. So and we designated the dark red color. How about this? How about this region and this region? How about the interaction between this region and this region? And also these two regions is located within this compartment, and also interaction will be happens like this. Okay, so usually the contact enrichment region is composed of enhancer or the promoter region. How about the border region? How about this border region? Okay, so this border region, we call it as a contact depletion region. This is a one TED. So this is, we call it as the TED border region. So this is we call it as TED border region. So TED border region, okay, let me see here. The interaction between this region and how about this region is medium. So that's why we designated pink color around here. So in contact depletion region is the composed of the several CTCF or cohesin and other factors. It's like this. Okay. Once again, heat map of TED shows the interaction frequency between the chromatin. So if interaction frequency is higher, we designated dark color. If the interaction is medium, we designated the pink color. If the interaction is not nearly happens, we designate the blue color. For example, the chromosome, the chromatin around here and chromatin around here, they are far away from each other, so interaction is not nearly happens. So we also divided by two regions. One region is a contact enrichment region, and the other is a contact depletion region. We call it as a TED region. Contact enrichment region usually is composed of the promoter and enhancer, and contact depletion region, we call it as TED border region. This region is composed of the CTCF and cohesin, and also other factors like this. Okay. So in this slide, I will explain the genetic disease due to TED disruption. So this is one example. The one chromosome is composed of the centromere region and telomere region. So between the centromere and telomere region, there are lots of the genes. For example, Wnt6 gene and IHH gene, and for example, EPHA4 gene and the PAX3 gene is like this. So each region make the TED structure, and each region make the TED structure. Each region make the TED structure, means that topological associating domain. So this is a border region, and this is a border region. So in one case is, for example, when deletion happen. For example, this region around this region is deleted. When the deletion happens, so it will disrupt this TED structure. So finally, it will exhibit genetic disease which has a short arm. Okay, in Korean we call it as a 단지점. Okay. How about another cases? When inversion, so when this location is inverted, or full length of this region is duplicated. Duplication happens, so it will induce genetic disease. We call it as F syndrome. So finger is fused each other like this. Okay. Another case is the part of this region is duplicated, or some region in this region is deleted. So in this case, it induces genetic disease which has the multiple fingers like this. So this kind of genetic disease is due to TED disruption. For example, when deletion happen, TED disruption TED structure will be disrupted here. Or when inversion happen, this TED structure will be disrupted. Or duplication happen, this TED structure will be disrupted. Or part of duplication happen, also this TED structure will be disrupted. Or some deletion happen, this TED structure will be disrupted. So in these cases, they make the genetic disease, this kind of genetic disease. So in this slide, I will explain the final subject, enhancer. So enhancer is cis-acting elements to interact with promote region. So this is the enhancer region, and this is promoter region, and this is our gene of interest. Okay, enhancer is a cis enhancer and promoter is located at same chromosome. That's why we call it as a cis acting element. Okay, so activate several activate bind to the enhancer region, so it changes the orientation. After that, or it recruits RNA polymerase. After that, the transcription can be actively happens like this. However, we cannot identify the enhancers very well because the enhancer is randomly scattered in many locations. So also it is almost impossible to find the enhancer due to lack of motif. Okay, so even though we find maybe this region will be the enhancer, but they don't have the motif, so we are not sure whether this region is an enhancer or not. However, epigenetics offer a breakthroughs to find enhancer. So people did several epigenetic analysis using several markers and found that enhancer is enriched with H3K27 acetylation. So this is a H3K27 acetylation. So enhancer region is highly enriched with H3K27 acetylation. Okay, let me see here. So this is the MYC gene. Okay, this is promoter region, and also they have enhancer region. Okay, so enhancer region is highly enriched with H3K27 acetylation, and also there is also another H3K27 acetylation, and also there is another H3K27 acetylation. And also people found that enhancer region is enriched with P300. So this is a P300. P300 is a histone acetyltransferase. So enhancer region is highly enriched with P300 like this. Okay. However, people found that enhancer is lacked with H3K4 methylation. So this is the H3K4. Okay. Another reason we will see that there are lots of H3K4 methylation. However, in this region, we found that H3K4 methylation is not nearly happens. Okay, so this is the especially the histone 3 protein is composed of several amino acids, especially and post amino acid is the lysine. K is lysine. So lysine, so post amino acid lysine residue is the 3-methylated. Methylated. We call it as H3K4 methylation. So we found that enhancer is lacked with H3K4 methylation. So even though there are several markers there, but we usually use these 3 markers. If you found that the H3K27 acetylation, or you found that P300 is enriched with this region, but however, if there is no H3K4 methylation, you will see that maybe this region will be the enhancer region. Okay. So in this slide, I'll explain the difference between enhancer and promoter. What is big difference? There are 3 big differences between the enhancer and promoter. First one is the H3K4 methylation. Okay, in the enhancer region, the H3K4 methylation is less. Okay, it's not nearly happened, but in the promoter region, you see here H3K4 methylation is highly happens. And also, a second difference is that direction of transcription. So enhancer region, the direction of transcription is bidirectional. However, in the promoter region, the direction of transcription is the directional. Only one direction. And also there is the length difference. For example, the enhancer region is usually the less than 4 kb. However, in the promoter region, there is no length limitation. Okay, this is a big difference between the enhancer and promoter. So distribution of H3K4 tree maturation, and also direction of transcription, and also length. Okay, so we have to know the difference between enhancer and promoter, especially focused on 3 criteria. Okay, learned Chapter 5, Epigenetics in Mammals, especially focused on DNA maturation and DNA dematuration, and the long non-coding RNA, and chromatin insulator, and TED. Finally, we learned enhancer. Okay, today we will finish the class.